The interplay between aging, autophagy dysfunction, and obesity is complex, and when biological sex is added to the equation, this complexity deepens further. In recent years, evidence has grown that males and females differ significantly in their metabolic trajectories with age, in how they accumulate and store fat, and in how their cells regulate processes like autophagy. These differences are not just superficial, but extend down to mitochondrial dynamics, hormonal signaling, immune function, and nutrient sensing. As such, the effects of nicotinamide mononucleotide (NMN) supplementation, a promising intervention for aging-related metabolic decline, may also diverge by sex, both in magnitude and in mechanism. Autophagy, the process of cellular cleanup and recycling, is critical to metabolic health in both sexes, but it is modulated by sex hormones in different ways. Estrogens are known to promote autophagy and mitochondrial quality control, particularly in the liver and brain, while androgens may have more complex, tissue-specific effects. With age, as estrogen levels decline in females and testosterone drops in males, these sex-specific regulatory layers are altered, changing the baseline upon which NMN acts. In this context, NMN's role as an NAD booster becomes especially intriguing because it acts upstream of multiple pathways, including SIRT1, AMPK, and FOXO transcription factors that are already differentially tuned by sex hormones. In studies involving aged rodents, a pattern has begun to emerge. Both males and females benefit from NMN supplementation, but the tissues most responsive and the degree of metabolic improvement appear to vary. For example, aged male mice tend to exhibit greater improvements in skeletal muscle insulin sensitivity and endurance capacity after NMN treatment, potentially reflecting their higher burden of mitochondrial dysfunction and inflammation in this tissue. In contrast, aged female mice often show more pronounced improvements in adipose tissue function, fat distribution, and inflammatory tone, perhaps due to the sharp loss of estrogenic protection after menopause that leaves white adipose depots particularly vulnerable to dysfunction. Autophagic activation also follows a sex-dependent pattern. In males, NMN robustly increases mitophagy and autophagosome formation in liver and muscle tissues, which aligns with improvements in lipid oxidation and glucose uptake. This is particularly important given that aged males are more prone to ectopic fat deposition and hepatic steatosis. In females, NMN appears to restore lysosomal function and reduce adipose inflammation more strongly, improving overall adipokine profiles and increasing the metabolic flexibility of fat cells. Interestingly, these sex-specific effects may also relate to differences in NAD, biosynthesis, and turnover. Females often display higher basal NAD levels in certain tissues, which may modulate their responsiveness to NMN supplementation. The hypothalamus, a central regulator of appetite and energy homeostasis, also exhibits sex-specific responses. Estrogen receptors in the hypothalamus modulate neuropeptide signaling, mitochondrial dynamics, and autophagic tone. NMN's ability to enhance hypothalamic autophagy may therefore interact more intricately with the female neuroendocrine system, possibly explaining why some studies suggest greater appetite normalization and circadian rhythm improvement in aged female mice than in their male counterparts. These differences extend to fat metabolism. Male animals often accumulate visceral fat more readily, which is closely associated with metabolic risk and autophagy impairment. NMN seems to counter this by restoring mitophagy and AMPK signaling in visceral adipose depots, an effect that is strongly associated with decreased insulin resistance in male models. On the other hand, female animals tend to store more subcutaneous fat and show higher susceptibility to adipose inflammation after menopause. NMN may be especially beneficial here by improving lipophagy, reducing macrophage infiltration in adipose tissue, and restoring anti-inflammatory adipokine profiles like adiponectin. What makes these findings especially promising is that despite the variation in tissue responses, both sexes ultimately benefit in terms of systemic outcomes: improved glucose homeostasis, reduced fat mass or fat toxicity, enhanced mitochondrial performance, and lower levels of chronic inflammation. Yet the path to these improvements is clearly modulated by sex-specific physiology, and understanding these nuances will be essential for tailoring NMN-based interventions in clinical settings. In human studies, sex-specific data is still emerging, but early trials suggest that both older men and women experience modest improvements in vascular function, physical endurance, and insulin sensitivity with NMN supplementation. Whether these benefits map onto the animal findings regarding autophagy is not yet fully known, but the growing recognition of sex as a biological variable in metabolic research makes it likely that future trials will include autophagic markers and stratify data accordingly. In conclusion, NMN holds therapeutic promise for age-related obesity and metabolic decline in both males and females, but the pathways through which it exerts its effects, particularly those involving autophagy, are not identical. Long-term metabolic improvements may depend not just on dosage or duration of treatment, but also on how NMN interfaces with sex-specific hormonal environments, tissue vulnerabilities, and cellular stress responses. Rather than seeing one sex as more responsive than the other, the emerging picture is one of complementary benefit. Males may gain more from NMN in skeletal muscle and liver metabolism, while females may experience greater repair in adipose and hypothalamic function. Understanding and leveraging these differences could usher in a more personalized era of anti-aging metabolic care, one where NMN is deployed not as a one-size-fits-all remedy, but as a precision tool aligned with the unique biology of the individual.